Oh man, you're recording.
I'll black it out so you can't see yourself.
I want to see that on YouTube later. We'll see. Well, maybe you should post it. We'll see how many hits we get.
You want me to post it? I'll post it.
Yeah, how many hits are we gonna get?
Everybody in here.
Oh,
I'll send out a link.
26.
I'll send out a link.
I'll click it twice so we get more hits.
Refresh it.
Actually, that would be interesting. You know, my mom would. She'll like email it to all of them. She'll be like, "This is what my daughter does. I understand now."
So conversations recorded.
You hear that, Mom?
Shout out.
Shout out to my mom, my bro. Yeah. All right, let's rewind. You can edit that. So I'm feeling much better today, thank you. So hopefully today's lecture will be much better, and we open the blinds so that we can get some light in. Okay, so what we have been talking about is how the Drosophila body plan is laid down, and we've talked about some set of genes called the parallel genes, and that they're expressed in alternating stripes. And so we've talked about gap proteins that are encoded by gap genes, and the local concentration of gap proteins control where the parallel genes are expressed. And this is a good example of spatial control of gene expression. Then in turn, the parallel genes lay down basically where the segmentation genes or segment polarity genes are expressed. And this results in defined boundaries between different segments. And we talked in particular about two signaling pathways, the Wnt pathway and hedgehog signaling pathways. And I'll review those again for us today. And how these two signaling pathways positively feed back on each between segments to really define the boundaries. Okay, so if we go back to looking at how the gap genes interact. So remember, the gap genes are the first genes that are expressed in the zygote, and there was hunchback was one of them, giant, Kruppel, and NERPs. So what this diagram is showing you is an embryo, and it's showing you where these different gap genes are located, or where they are expressed in the embryo. So we see that the gap gene giant is expressed in the anterior, as well as in this section in the middle. Hunchback is expressed in very specific locations. NERPs, or sorry, Kruppel, rather, in purple, and NERPs are right in the center. So part of the reason why these gap genes are expressed where they are is because they have negative feedback amongst each other. So for example, hunchback and NERPs, they negatively feedback each other's expression. So wherever hunchback is, you won't see NERP expression, and that's what you see here. And where NERPs is located, you don't see hunchback because it blocks its expression. So remember, these are transcription factors, and these transcription factors regulate the expression of genes. And so wherever hunchback and NERP proteins overlap, they'll inhibit the other's genes in that location. And it turns out there's a similar relationship between giant and Kruppel. So this helps set up these very localized expression patterns in the embryo. And this starts at the beginning with the gap genes. Okay, so you end up with an embryo that already has unique domains that contain different combinations of transcription factors. Okay, and so last time we talked about how the parallel genes are responsive to these different local concentrations of gap proteins. And so I showed you that the regulatory regions in the DNA for even skipped, or EVE, which is a parallel gene, each have a different set of activating and repressor binding regions, and that the gap proteins are the proteins that bind to these activating or repressing regions. And so, for example, for stripe 2, where Eve is expressed in stripe 2, there is high concentrations of hunchback, lower concentrations of bigoid, no concentration or zero giant, and zero purple. So if we look at the regulatory region for Eve in this stripe, all the repressors, giant and purple, they're not present, so they're not binding here. And there's a lot of hunchback, and there's low concentration of bigoid, but there are multiple places where bigoid can bind and activate the Eve gene in this stripe. And so these are the DNA regulatory regions that are responsible for Eve expression in stripe 2. Okay, so if we look at this using a reporter gene, this really, you can really see how specific these regulatory modules are. So if you hook up the stripe 2 module, which I just showed you, to a LAC-Z reporter gene, and then you do immunohistochemistry, you'll only see LAC-Z expression where stripe 2 is supposed to be. But if you do immunohistochemistry for the Eve protein, you'll see all the stripes because this IHC can't distinguish whether you want to look at Eve in stripe 2 or stripe 4 or stripe 5. It's just looking for the final product. Okay, and so we also know that each stripe's regulatory region is very specific for a location, because experiments like this have been done, where this is basically the same diagram that I showed you here and here from a different textbook. So here you have stripe enhancer regions for stripe 2 and stripe 7, stripe 3, stripe 4 and 6, and stripe 1 and 5. Okay, so if we take just this enhancer region and we take the enhancer region for stripe 1 and hook it up to a LAC-Z reporter, and then do immunohistochemistry for LAC-Z, and after we put this into a wild-type embryo, we only see staining of LAC-Z in stripe 1. If we take now the stripe 5 enhancer and we put that in front of a LAC-Z reporter, we only see staining in stripe 5. And then if we can make combinations of it, if we stick stripe 1 and stripe 5 enhancers together, put them in front of LAC-Z, we see staining in stripe 1 and 5. So these type of data show you that the regulatory regions are very tightly controlled in their location. And if the gap genes are not expressed quite right, then it messes up the whole concentration of local gap proteins that affect the pair role gene expression. So here, where we put stripe 1 and stripe 5 together under the LAC, and then look for LAC-Z, in an embryo that is missing giant expression, you see the band of staining in stripe 5 is more diffuse and it's wider. And that's because the giant is missing to create the local concentration of gap proteins that are specific to inhibit this staining in the wild-type embryo. Okay, so this shows how the gap proteins and the pair role genes interact. Any questions on this? Still thinking? Okay, so the pair role genes are expressed in stripes, as I said. And so there's Eve in blue and Fushi-Toratsu in brown. And I read yesterday that Fushi-Toratsu in Japanese means too few segments. Okay, so it makes sense. It's basically the phenotype of the embryo that is missing